Now we're going to talk about glycolysis. Glycolysis is the anaerobic breakdown of glucose to pyruvic acid, which makes a small amount of energy in the form of ATP. So first we're going to go through all the steps of how to get from glucose to pyruvic acid, and then we're going to talk about what's used and produced. So first we start with glucose, which we know is a 6-carbon sugar. Then we take 2 ATP, and then it becomes 2 ADP, and that lets us produce a 6-carbon compound. It is phosphorylated, as we call it, where it has phosphate groups on each end. So each of these circles represents one of our carbons. Now that is changed into 2 molecules of G3P, and essentially that just is what we had previously broken into the 2 halves. The next step, we add 2 phosphates, and we have 2 NAD plus become 2 NADH plus 2 hydrogen ions, and that gives us this, which is those same groups but just phosphorylated more. And in our next step, we're going to take 4 ADP and turn those into 4 ATP by removing those phosphates from the ends. And that's going to give us just our carbons left here, plus some other things we're not showing for simplification. And that is our 2 molecules of pyruvic acid, which we'll use later on in other respiration processes. So now let's look at what's used and what's produced. If we go through our list, we can see that we use 1 molecule of glucose to start. We use 2 ATP in our beginning, and we use 2 NAD plus. We end up producing 4 ATP, which will give us an overall net of 2 ATP produced from this process since we used 2. We're going to have 2 molecules of pyruvic acid. We're going to have 2 hydrogen ions, and we're going to have 2 NADH. And this is the cycle of glycolysis, which is one of the most fundamental types of cellular respiration since it's anaerobic. Bacteria even will carry this out to give energy.